In October 2023, a scientific breakthrough that went largely unnoticed by the public fundamentally changed our understanding of life's beginnings. Researchers at the Weizmann Institute of Science in Israel accomplished what many considered impossible: growing mouse embryos completely outside a natural womb, from fertilization to full term. This remarkable achievement opens unprecedented possibilities for science, medicine, conservation, and raises profound ethical questions about the nature of life itself. Join us as we explore this revolutionary technology, its challenges, applications, and the far-reaching implications that extend well beyond reproductive science. The groundbreaking research led by stem cell biologist Jacob Hanna at the Weizmann Institute marked the first time mammals have been grown entirely outside a natural uterus. Published in the prestigious journal Nature, this achievement represents a paradigm shift in developmental biology. The team cultivated mouse embryos from fertilization to full term using a specialized rotating vessel system where the embryos developed normally, with fully functioning organs and limbs. What makes this technology truly revolutionary is the specialized pressurized rotating vessel filled with nutrient-rich fluid that precisely mimics the conditions of a mouse uterus. The system continuously monitors and adjusts oxygen levels, nutrients, and pressure in real time, creating a dynamic environment that supports complete embryonic development. Unlike previous artificial womb experiments, this breakthrough encompasses the entire developmental process without requiring transfer from or to a living animal at any stage. The Weizmann team's success came after overcoming several critical challenges that had prevented previous attempts. First, they developed a synthetic endometrial interface that recreates the complex biochemical signaling required for implantation, a process previously thought to require living tissue. This artificial surface mimics the intricate molecular environment of a natural uterine lining, providing the necessary signals for the embryo to attach and begin development. Second, they implemented an advanced perfusion system that gradually changes the nutrient composition to match each developmental stage. In natural mammalian reproduction, this happens through changes in maternal-fetal circulation, but the team recreated this process artificially with remarkable precision. Third, they solved the mechanical challenges of accommodating rapid embryonic growth by designing an expandable chamber with variable pressure gradients. This innovation allows the artificial environment to adapt as the embryo grows, providing the appropriate physical conditions throughout development. This technology creates unprecedented opportunities to study mammalian development with complete environmental control. Scientists can now observe every moment of development without invasive procedures, potentially answering fundamental questions about how genetic instructions unfold into complex organisms. For the first time, researchers can precisely manipulate individual variables during development to understand their effects. This could revolutionize our understanding of developmental abnormalities, which affect approximately 3 to 5% of human births worldwide. Conditions that have remained mysterious despite decades of research might finally be understood by observing development in real time and under controlled conditions. The ability to witness the entire developmental process also opens new avenues for studying the effects of environmental factors, medications, and genetic variations on embryonic development, potentially leading to new treatments and preventive measures for developmental disorders. While the mouse breakthrough represents a fundamental proof of concept, scaling this technology to larger mammals presents unique challenges that researchers are tackling through unexpected approaches. A collaboration between the University of Tokyo and biotechnology firm Emulate has developed organ chip technology that recreates the maternal-fetal interface at the microscale. Their placenta-on-a-chip system successfully replicates the complex exchange mechanisms between maternal and fetal circulation for larger mammals, including pigs and sheep. Engineers from ETH Zurich have taken an industrial approach, adapting principles from bioreactors to create modular, scalable artificial gestation systems. Their approach incorporates microfluidics and biomimetic materials, allowing for potential expansion from small to large mammals. These systems use synthetic membranes with gradient porosity that mimic the selective permeability of natural placental barriers, enabling precise control of substance exchange at different developmental stages. Researchers at the Beijing Genomics Institute have taken a different approach by focusing on computational modeling of species-specific developmental requirements. Their machine learning algorithms analyze thousands of parameters from natural pregnancies across different mammal species to predict the precise environmental conditions needed for artificial gestation. This data-driven approach has already yielded surprising insights about previously unknown differences in metabolic requirements between closely related species during gestation. One unexpected challenge emerged when researchers attempted to scale the technology to medium-sized mammals: the problem of waste elimination. Natural mammalian development relies on maternal systems for removing embryonic waste products, a process that becomes exponentially more complex as embryo size increases. A University of California, San Diego team has developed a novel biofilter system using genetically modified bacteria that selectively process embryonic metabolic byproducts without disrupting developmental signaling molecules, an elegant solution inspired by symbiotic relationships found in nature. The rapid advancement of artificial womb technology has outpaced ethical frameworks, creating a regulatory vacuum. The International Society for Artificial Reproductive Technologies, formed in early 2023, has proposed the developmental potential scale as a new framework for evaluating synthetic gestation systems. This scale measures not only technical capabilities but also addresses the complex moral considerations of creating life outside natural processes. Unlike previous bioethical approaches that focused primarily on human applications, this framework incorporates perspectives from evolutionary biology, animal welfare science, and indigenous knowledge systems about the relationship between birth and ecological systems. It acknowledges that artificial gestation represents not merely a technological innovation but a fundamental shift in how life comes into being. The World Health Organization has established an International Advisory Committee on Synthetic Reproductive Technologies, which released preliminary guidance in September 2023. The report recommends distinguishing between therapeutic applications, addressing infertility or pregnancy complications, and enhancement applications that modify or optimize development, with different regulatory approaches for each category. Religious scholars across traditions have begun re-examining theological perspectives on the nature of birth and creation in light of these technologies. The Vatican's Pontifical Academy for Life convened an unprecedented interfaith symposium in December 2023, bringing together Islamic, Jewish, Hindu, Buddhist, and Christian scholars to discuss the spiritual dimensions of artificial gestation. Despite significant theological differences, the symposium produced a surprising consensus document acknowledging that the moral status of artificially gestated beings should be considered equivalent to naturally gestated ones. This position could significantly influence future regulatory frameworks as it provides a spiritual foundation for treating artificially developed life with the same ethical considerations as natural life. The symposium also explored deeper questions about the spiritual significance of birth processes across different faith traditions and how these new technologies might be integrated into existing religious frameworks concerning the beginning of life. The ripple effects of artificial womb technology extend far beyond reproductive science. Conservation biologists have identified potential applications for endangered species preservation. The San Diego Zoo Wildlife Alliance has initiated a program to explore artificial gestation for critically endangered northern white rhinos, with only 2 females remaining worldwide. The program aims to use stored genetic material to potentially revive the species without requiring surrogate mothers from related rhinoceros species. This approach could revolutionize conservation efforts for species on the brink of extinction, especially those where limited numbers of females represent a reproductive bottleneck. By using artificial gestation, conservationists could potentially increase reproductive rates beyond what would be possible naturally, accelerate genetic diversity preservation, and even revive species using preserved genetic material when natural reproduction is no longer possible. In developmental pharmacology, artificial gestation systems provide unprecedented opportunities to study how medications affect developing organisms without harming pregnant animals. Researchers at the University of Pennsylvania have begun using simplified versions of these systems to test how various pharmaceuticals cross the placental barrier and influence organ development, potentially revolutionizing prenatal medication safety testing. This could dramatically reduce the risks associated with medication use during pregnancy by providing detailed information about developmental effects before human clinical use. Perhaps most surprisingly, artificial womb technology has influenced fields seemingly unrelated to reproduction. Bioengineers at Stanford University have adapted the nutrient delivery systems developed for synthetic uteruses to create new approaches for cultivating artificial organs for transplantation. The precision environmental control mechanisms developed for artificial gestation have proven ideal for growing complex human tissues and organs outside the body, potentially addressing the critical shortage of transplantable organs worldwide. The precision environmental control mechanisms have been adopted by researchers developing self-sustaining habitats for potential use in space exploration, demonstrating how innovations in one field can catalyze unexpected advances in others. The technology has even inspired novel approaches in sustainable agriculture. Aquagenesis, a Netherlands-based startup, has adapted artificial gestation principles to develop sealed hydroponic systems that create perfectly controlled environments for plant growth. Their seed-to-harvest enclosed systems use 97% less water than traditional agriculture while eliminating the need for pesticides, potentially transforming food production in water-scarce regions. These systems apply the same principles of precise environmental monitoring and adjustment that make artificial gestation possible, but for plant life instead of animal development. The closed-loop nutrient recycling developed for artificial wombs has proven particularly valuable for creating sustainable agricultural systems that maximize resource efficiency while minimizing environmental impact. As artificial gestation technology advances, we stand at the threshold of profoundly reimagining one of life's most fundamental processes. The coming decade will likely see attempts to extend these technologies to primates and potentially humans, raising complex questions about the relationship between technology and natural processes. The boundaries between therapeutic applications, helping those who cannot carry pregnancies, and enhancement uses will require careful navigation by scientists, ethicists, and policymakers alike. What remains clear is that artificial gestation technology represents more than a scientific curiosity. It constitutes a fundamental shift in our relationship with reproduction itself. As we develop the capacity to bring life into the world through entirely new pathways, we must simultaneously create new ethical frameworks, regulatory approaches, and cultural understandings that honor both the remarkable potential of these technologies and the profound responsibility they entail. The possibilities range from addressing infertility and pregnancy complications to preventing genetic diseases through controlled development, but each advance will require thoughtful consideration of its implications for humanity's relationship with nature and technology. The complete growth of mouse embryos outside a natural womb by the Weizmann Institute represents one of the most significant yet under-recognized scientific breakthroughs of our time. This technology has evolved from a specialized research tool into a catalyst for innovation across multiple fields, from conservation biology to sustainable agriculture, from pharmaceutical testing to space exploration. As researchers overcome the challenges of scaling to larger mammals through computational modeling, bioengineered solutions, and interdisciplinary approaches, we move closer to applications that could transform human reproduction and medicine. Yet this progress occurs within a complex ethical landscape that religious leaders, bioethicists, and policymakers are only beginning to navigate. The artificial womb technology represents more than scientific advancement. It fundamentally changes our relationship with one of life's most profound processes. The story of artificial gestation is just beginning, but it already promises to reshape our understanding of life's earliest chapters while challenging us to develop ethical frameworks that match the power of this revolutionary technology. As we stand at this threshold, the responsibility falls to scientists, ethicists, and society at large to ensure these remarkable capabilities serve humanity's highest values while respecting the wonder and complexity of life itself. Thanks for watching. Please like, subscribe, and hit the bell icon for updates. Your feedback is appreciated. We hope to see you again.